This is the lecture for cell structure and function. One thing that I will go ahead and tell you or warn you about is there are 113 slides. I just skipped through some of them, but it's still a lot. And we really don't need to skip around a lot because this material is often on the TEAS test along with the information on chemistry. So you really need to be at least educated in cell structure and function before you take that TEAS test. And a lot of you or some of you have not had biology where you would have learned this information. So I try to go through it slowly enough that that you could get something out of it. So the four basic concepts of the cell theory. This entire chapter is based around the cell theory. Cells are the building blocks of all plants and animals. Cells are the smallest functioning units of life. Anything smaller than a cell is not considered a functioning unit of life. Cells are produced through division of pre-existing cells. And each cell maintains internal homeostasis. Cells in the human body. The human body contains trillions of cells. So let's say that the human body contains 100 trillion cells. 90 trillion of those cells, 90% of those cells are actually not human cells. They are microorganisms that live on our skin or inside of our bodies. And that sounds disgusting, but it's not. We need those microorganisms to help digest our food, to help produce vitamins. They have many functions, and we could not live without them. Homeostasis of the body is maintained by coordinated action of cells. Cells communicating and coordinating their actions. And then, of course, cells come in a variety of shapes and sizes. The study of cells is called cytology. Cyto means cell. Logy means study of. We study cells using light microscopes, which is what you find in a typical college biology laboratory, and electron microscopes. There are two types, the transmission electron microscope and the scanning electron microscope. These are both not normally found in community college laboratories, sometimes not even at the university level, but they see detail a whole lot better than the light microscopes do. They can magnify much greater, to a much greater extent. You can see here that the shapes of cells are as varied as the type of cells. This is an egg cell or an ovum. This is a fat cell. As you can see, its function is to store fat. All that yellow is fat. The nucleus of the cell is actually squeezed up against the plasma membrane. A neuron has long branching structures for transmitting impulses. This is a bone cell. Blood cells are rounded. This is a smooth muscle cell, like you would find lining your blood vessels. And these are cells that line your intestinal tract. This is a goblet cell that produces mucus to line your intestinal tract. And these structures are called microvilli, and they absorb the nutrients that your intestines break down from the food you eat. So that's a wide variety, but I just touched on a few cell types. We have many more. All cells, no matter what type, all of them have a plasma membrane, or you can call it a cell membrane. I tend to call it a plasma membrane because that's what I've been calling it for years. So that's usually what I'm going to call it, but cell membrane is just as appropriate or correct. The plasma membrane separates the cytoplasm, which is everything inside the cell, from the surrounding environment, which typically is an extracellular fluid, except for the cells on the outside of your body, your skin cells that are exposed to the atmosphere. The only time they're surrounded by fluid is, for example, if you go swimming or take a bath or a shower. But most of our cells are surrounded inside of our body by fluid, and we call that extracellular fluid. This is a figure of a typical animal cell, and my view of it is very difficult to see, so you may want to go back to your slides and blow it up to see everything more clearly. But I will go through some of these organelles with you and explain their function. This is the surface of this particular cell. It's covered in microvilli, which are folds of the plasma membrane, microscopic foldings of the plasma membrane. And the function of microvilli in intestinal cells and anywhere else that you find those is always for the absorption process. So these microvilli would absorb the nutrients that are broken down from the food we eat, and they would absorb them into our bloodstream. This is the nucleus, which contains the DNA or the genetic material. This, of course, is the plasma membrane. The cytoplasm is everything inside, and here's another word, cytosol. The only difference between cytoplasm and cytosol is just the fluid. Cytosol is the fluid that you find between the plasma membrane and the nucleus. And the cytoplasm is just everything inside the plasma membrane. The cytoskeleton is highlighted here. You see fibers. You see different protein fibers scattered throughout the cell. They have different jobs. There's some up here near the microvilli, but these fibers have different jobs. They help maintain the shape of the cell. They help keep the organelles in place, the organelles like the nucleus, and they can help with movement inside the cell. Let's see, the centrioles are these two structures here, and they are used when the cell divides, they're used in cell division. Some other structures I can see, this is the mitochondria, which produces ATP. This is where energy is produced in the cell. This is the Golgi complex, or a Golgi apparatus or Golgi body. I've heard it said many different ways, but just make sure you know it's the Golgi. These channels that are studded with dots, that are covered with dots, are called rough endoplasmic reticulum, rough ER, and they function to assemble proteins, and mainly they function in the synthesis of proteins. And then there is the smooth ER that's outside of the rough ER, and that functions mainly in the synthesis of lipids. And here we have, let's see, sometimes you will see cilia covering the surface of a cell. These are little hairs that help move some type of fluid or move something across the surface of the cell. There are in a man's testes, there are cells that contain cilia that help move the sperm along before they are able to swim on their own. And what we see here is the Golgi apparatus, which functions in modifying, altering, storing, and packaging the products of the endoplasmic reticulum, the rough and the smooth ER. trying to see if I could blow it up, but I can't. This looks like proteasomes. Okay, so these contain enzymes that break down proteins. Ribosomes are found in all kinds of cells, no matter what kind of cell it is. Ribosomes are represented as little dots. They are not surrounded by a membrane, but they can be free floating in the cytoplasm, and they can be attached to the rough ER. They do synthesize proteins. Peroxisomes are represented as purple vesicles, and they help to break down fatty acids. Lysosomes are represented as green vesicles, and they help to break down many things, including the nutrients from the food that we eat. Here is the nucleus. When the cell is not dividing, the DNA is called chromatin, and the membrane that surrounds the nucleus is called the the nuclear envelope, and I went too fast. And then in here, this is a nucleolus, sometimes one and sometimes more than one. The nucleolus produces ribosomes. I already mentioned to you about the mitochondria and the endoplasmic reticulum. The cell theory was developed over many years. What are its 4 basic concepts? Okay, well, we can just go right back to the first page. Cells are the building blocks of all plants and animals. They're the smallest units of life. That they are produced when pre-existing cells divide, and each cell maintains homeostasis. The study of cells is called cytology. Four general functions of the plasma membrane. The plasma membrane provides the cell with physical isolation from the extracellular fluid. It regulates exchange with the environment. Some particles are able to pass through the plasma membrane while others are not. It allows for sensitivity to the environment. That, sometimes our cells need to be able to read chemical signals from other cells or from the environment. And they provide for structural support. For example, muscle cells and bone cells. The structure of the plasma membrane includes lipids, proteins, and carbohydrates. The lipids are phospholipids and cholesterol, which here is referred to as steroids. But phospholipids form, let's see, here we go. The phospholipids form a double layer, and that forms the backbone of your plasma membrane. The tails of the phospholipids are hydrophobic, and then the heads, these red heads, are hydrophilic, meaning they can be in contact with the watery environment outside the cell and inside the cell. You can see the cholesterol molecules, and you can see on the outside of the cell carbohydrate chains of glucose. These help with cell recognition. For example, if this was a red blood cell from someone who had type A blood, the molecule would be called an A antigen. The molecule that showed that it was type A would be called an A antigen. This is also what is used in finding someone with tissue compatibility. If you are going to get an organ transplant, finding a compatible donor, these carbohydrate chains. So they identify the cell. Then the blue large structures are various proteins that are in the plasma membrane that have different functions. Phospholipids are the major component of the plasma membrane, and cholesterol gives the plasma membrane a certain stiffness, so it's not so fluid. Lipid-soluble materials easily cross through the